no ar.
Olá, pessoal, boa tarde. Desculpem o pequeno atraso, a gente estava resolvendo algumas pendências aqui tecnológicas, mas agora conseguimos fazer com que todo mundo esteja aqui. E gostaria de agradecer ao sucesso que foi a semana passada, o Oscar da Biologia. Foi muito legal. E hoje, então, nós temos uma palestrante do exterior, do Canadá. Então, nós temos hoje a Charlotte Coates, que é aluna de mestrado do professor Peter Kevan, e ela vai contar, então, um pouquinho para a gente sobre a termorregulação em caules de planta. Hoje é mais ainda temático essa foto, porque essa foto tem tudo a ver com o trabalho lá do Canadá, que eu participei de um começo lá. Então, eu gostaria de agradecer a Charlotte, e eu vou, então, agradecer para ela em inglês agora. Depois vocês podem deixar as perguntas aqui no chat, e pode ser em português ou em inglês, e aí no final a gente conversa. So, Charlotte, I was presenting you and telling them that I'm very grateful for you to be here with us today, and also Peter for joining us, and I hope we have a very good discussion after your talk. So thank you very much for your time, and I'm very glad I will see the new results on the project. So now you have the word.
Okay, thank you so much, Patricia. Let me just share my screen here. Okay, how is that? Can everyone see it? Yes? Okay, great. Okay, so thank you so much, Patricia, for inviting Peter and I today to present. It's great to have this opportunity. So our presentation today is titled, Do Herbaceous Plants Regulate Their Temperatures? And presented by myself, Charlotte Coates, and I'm part of Peter Kevan's lab at the School of Environmental Sciences at the University of Guelph. And this work is funded by the COHA, which is called the Canadian Ornamental Horticultural Alliance, and we're very grateful for their funding for this project. This is the third year of our project at the University of Guelph. So this is a photo from our first field season back in July 2018, and you probably recognize this is Patricia and I doing field work on squash in Peterborough. So this was the start of the work, but we have come a long way since then. And I'm very grateful to the project and to Patricia for teaching me a lot about plants and pollinator interactions, as well as experimental design. Okay, so what are we going to talk about today? Today we'll talk about what is micrometeorology, and then we'll talk about how heat is produced by metabolism in plants. Next we'll talk about passive solar heating in plants, the microgreenhouse effect, the methods to measure this effect, some examples of microgreenhouses or plants that could be considered microgreenhouses. And then I'll discuss some of the results so far and some of the relationships that the temperature work that we're doing has to plant and pollinator relationships. So what is micrometeorology? Well, endomicrometeorology is a very large word, but we can break it down into smaller parts. Endo means within, micro means very small, and meteorology means the study of the climate. So we know that micrometeorology is the study of the climate near the ground. And the field of micrometeorology around plants is actually fairly well studied, especially in controlled environments. Here in Canada, we have a lot of our food being produced in greenhouses, so they're able to control a lot of the environmental conditions within their growing habitats. So some of the things that people will look at when they're studying micrometeorology is the wind, the atmospheric movements, light conditions and radiation, temperature and heat, humidity, moisture, and precipitation, and how all of these factors must be maximized in order to have healthy plants, depending on whatever your plant it has the need for, then you can somewhat control the environmental conditions around that if you're able to in a greenhouse. Or in the field, the technology is increasing all the time to have better environmental monitoring, and these things are used to inform decisions about when to plant seeds, when to watch for pests, when to harvest, and things like that. So given these new abilities that we have with environmental monitoring, we now ask the question, what micrometeorological conditions exist within the plants themselves? Well, what do we know about how to answer this question already? Not much. We don't really know what micrometeorological conditions exist within plant parts. We know that a lot of plants do have hollow structures, flowers, stems, fruits, and galls, but this is little studied until our project began in 2018. We know that these plant parts enclose air around them, so we consider them to have separate atmospheric conditions than the outside of the environment. It is also known that, that the assumption is that plants always take on the temperature of their environment. Furthermore, to avoid overheating, it is known that transpiration in leaves causes water evaporation, and when the water is leaving the plant, this acts as a cooling mechanism for the plant to keep the plant tissue cool. However, we also know that some plants actually produce heat to benefit their survival in some way. So these are some of the things that we already know about conditions within the plants, but we can go even further than this. So firstly, we're going to talk about metabolic heating. So metabolic heating basically means heat that is being produced by the plant through some sort of metabolic process. And the function of this heat could be to keep the reproductive organs of the plant warm. In the photo on the right, you can see that in the center of the image, the red section is the male part of the flower's reproductive organs. And this area is almost 20 degrees warmer than the leaves. And this is a huge difference. And we can see that this difference in temperature causes the snow to melt here in Canada in the springtime. This plant, by the way, is called the Eastern skunk cabbage. And the metabolic heat has been well known for a long time and has been studied since the 1700s, along with other Irradiances species. Another example of metabolic heating is this beautiful flower, the sacred lotus. And recently, only in the past few years, the exact mechanism that causes heat was discovered in the sacred lotus plant. So heat is produced when the temperature drops, the rate of oxygen consumption increases, and this process happens on a cellular level, as you can see on the right side of the screen, where you can see the cellular respiration is increasing the plant in response to the drop in temperature. And this process is probably similar to that happens in the skunk cabbage that I just shown, and also in other lilies. And again, it is hypothesized that the heat is produced to protect the plant reproductive organs, as this process kicks in when the plant is experiencing colder temperatures. And this, again, is a mechanism that happens on the cellular level. Another example that we've recently discovered this summer is that heat production can also occur when nectar is being produced. So in this image, you see on the left side a squash flower, and on the right side, you see another squash flower that's taken with a thermal image. And you can see the ring around the center of the flower is about 3 degrees warmer than the petals. Now, why does this occur? Well, nectar production in the flower occurs after the sun rises in the morning from about 7 until 11 at night, 11 in the morning. And when the nectar is being produced, there's a process of converting the starches to the sugars to produce the nectar. And this process needs enzymes to make it happen. And the enzyme activity actually produces heat as a byproduct. So we wonder, could this also be a cue for pollinators in some way? We know that the heat is being produced within the flower, and that bees actually have a different spectra of light that they can see than us. So when a flower is producing heat, bees may actually be able to sense that and use that as a cue of which flower they might choose. So this summer, I was able to collect some data on the floral temperatures and floral visitation to see if any trends might appear with bee preference. Right now, the results show that all of the flowers, no matter what variety, have a similar heat differential in this time of day. So we haven't actually observed any differences in floral visitors because all of the flowers have this phenomenon, but it's still an interesting question when you look at how pollinators and flowers have evolved together. So next we'll talk about some other modes of heating in flowers that aren't metabolic. The plant's not producing heat itself, but it's collecting it passively. And this was first studied in the 20th century. And recently, there's been a book that has been published called The Thermal Ecology of Flowers that does a really excellent review of this. And that's by Dr. van der Kooi, and Peter Kevan was also involved with that publication. So the different types of flowers, there are 5 different types. So the first type, number 1, is the simplest design. It's a disc blossom. So examples are dandelions or sunflowers. And the second type that can collect heat from the sun, a bit more complex, is a dish or a bowl, number 2. And some examples of these are poppies, avens, anemones. The third type is pubescence, so the role of hair or fuzz that leads to heat absorption and trapping of heat, like if you're wearing a nice warm sweater. Some examples of these are willow, catkins, or just any other fuzzy plant. We have a lot of fuzzy plants in Canada because it's really cold here. Number 4, we have convectional heat trapping. So these are the bell-shaped flowers. We have hanging bell-shaped bluebells and heather that have a similar shapes that can collect heat. And number 5 is hollow flowers that act as microgreenhouses, such as snapdragons, loseworts, or campions. So we're going to go into all of these structures a little bit more into detail so we can see how do they actually collect heat from the sun. So firstly, we'll talk about the disc blossoms. So it's well known that sunflowers all face the same direction in a field, and this is due to diaheliotropism, aka they track the sun. They follow the sun as it moves across the sky, changing the floral direction to get the maximum solar exposure. Now the temperature, you can see on the left side of the screen, follows a cosine relationship to the angle of the sun. So at a 90-degree angle, when the sun is straight above, the temperature is highest, about 3 degrees warmer than the air temperature. In this graph, we can see that. That's the peak of the cosine function. And when the angle is at 0, when the sun is on the horizon, the temperature difference between the flower and the air is at about 0. So we can see that the flowers track the sun and they follow a cosine, the temperature follows a cosine relationship. This is another example of a disc flower, is dandelions. Now, dandelions are considered a pest plant, and in Canada, a lot of people go to very extreme measures to remove them from their lawns because they don't like them, as they're extremely fast growing and they have this amazing seed dispersal mechanism, even in cold temperatures at the end of winter. So how do dandelions succeed in reproducing so fast when there's not that much sun, there's not that much warmth in the air for them to gather? Well, we can see from this image, that's again an infrared image, the same, so one is the infrared image and one is the paired color version. And you can see the little blue and red triangles. Those are where I've created boxes to get the temperatures from each of the flowers. And we found that the mean surface temperature of the flowers is about 24.8 degrees, and the surface temperature of the surrounding grass is about 22.3 degrees. So there's a difference of about 2.5 degrees between the flowers and the grass, which is quite interesting because you would always expect a plant to have the same temperature as the environment. But as we know, that because the dandelion is a disc shape, it can actually attract more heat and preserve that heat. And then maybe that helps it to then have this fast growth rate and the seed dispersal mechanism. The next type of flower is an open bowl blossom. So open bowl blossoms, they're similar to the disc blossoms. They have a similar shape, but they're just more curved in. They can also be diheliotropic, like the Arctic poppy here. We see that. And so these flowers follow the sun. They have a similar pattern of peaking. The temperature peaks when the sun is at a 90-degree angle. So here we see the temperature difference between the flower and the air is more than 5 degrees. So that is quite significant, especially when you consider that this is in an Arctic region. So they really need to collect as much heat as they can. So you can see that plants have evolved different strategies to do that. Another way that we can conceptualize this is by looking at how the flower performs when we remove certain organs to see which parts are actually responsible for creating this temperature difference. So on the left screen, we can see that the whole flower has the highest temperature difference between the air at about 6.5 degrees. If you remove the sexual organs, it's lower at about 4 degrees. And without the petals, the flower is more similar to the air temperature, just slightly above. So we can see that the petals play a large role in reflecting the sun back into the center of the flower to concentrate that heat and keep that heat and hold on to it in order to ensure the success and reproduction of the plant. Now, this phenomenon is well studied. We see the reference from Greek mythology where Clyte, who is she's in love with a sun god named Helios, and her neck is twisted because she follows the sun god around, looking at him just like these flowers do. So this phenomenon has been well observed for many centuries. Next, the temperatures within pubescent flowers. So the example here is Willow, Salix, Catkins. And I've shown the picture of the mittens because the hairs act like mittens to keep the plant warm, to insulate it from the cold temperature. We can see that in general, the plants are about 2 to 4 degrees warmer than the air when the air temperature is around 5 to 10 degrees Celsius. So this is a very cold ambient air temperature, but the plants are still able to elevate their temperature above that, which is really interesting. The sexual differences between these plants is also interesting, that the pistillate, or the female willow flowers, are warmer than the male flowers by about 1 to 3 degrees. And now this clearly can play a role in how the plant is able to ensure the viability of its offspring and the viability of the pollen, and ensuring that reproduction can take place. Now, the orientation of the flower also plays a role in the temperature regulation, as the insulated side, whatever side has the sun shining on it, is going to be warmer than the other side by about 1 to 2 degrees. And in cloudy weather, well, unfortunately, the floral temperatures, the flowers aren't able to get any extra heat from the sun. They're just the same temperature as the ambient air. So you can see that when it is sunny, the flowers have these mechanisms in order to harness as much of the sun's energy as possible. And another thing to think about with the hairs is a different way to conceptualize it. In the heat transfer, it's sort of increasing the boundary layer. When you look at like Fourier heat law, if the boundary layer increases, then the amount of time for the temperature to change also increases. And now the last type of flower that we'll talk about, the second last type is a hanging flower shaped like a bell. So the examples we have here are Ericaceae, and they were around 1 to 2 degrees warmer than the surrounding air, the white flower. And others, Mertensia campanula, may also function similarly, but it has not been yet investigated. And the diagram in the bottom, you can see how the convection works. So convection is the heat transfer through molecules, through fluid dynamics. So you can see that the hot air moves up and displaces cold air currents. And yeah, convection teaches us how heat can diffuse through a space. And the bell-shaped flowers are really perfect for collecting that warm air that is rising from the earth that has been warmed by the sun, because soil is dark, it absorbs a lot of the sun's heat. And then the flowers can harness that heat that the soil has absorbed as it rises up into the atmosphere. So another strategy that plants have. Okay, so now we'll discuss this a little bit more in detail. So open flowers, we know that the reflection of the light of different wavebands, including infrared or the heat-producing waves, this is the mechanism that heats up the flowers. So the sun shines down onto the different shapes or forms that we have visualized here. And you can see that different shapes will reflect the sunlight differently. In the bottom left, you see how a bowl-shaped or a more disc-shaped flower will compare to each other. A disc shape is more flat. You see on the left is a bit more flat. So the sun, it's sort of focused here, but some of it is lost. If you have a more curve, a bowl shape, then the sun is actually going to the radiation is actually going to concentrate in the center more, and that's why we saw in the other slides that the bowl-shaped flowers had actually some of the higher temperature differences between that and the air. The open bells and pubescent flowers, they interact differently with the sun. But we know that in all plants, it's the cell pigments that reflect and absorb light, just like we know about chlorophyll. So the pigments are in solution or in plastids, or in both. And these placements can vary depending on the flower and the cell layers. So this differential placement of pigments would reflect the light differently. Another thing to consider is the surface texture of the flower. Is it glossy, is it matte, or is it iridescent? Is it reflective a little bit? And there could be other mechanisms that could be coming into play with this as well. So this is discussed in detail in the thermal ecology of flowers that was published just last year. So I would encourage you all to check it out if you're interested, or yeah, we can have more questions at the end of this presentation as well. Okay. And now the last type of flower that can passively absorb heat from the sun is a microgreenhouse flower. And this is also exists in stems. So we know that solar radiation is trapped inside a greenhouse, just like we see on the left. And this enables the sun's energy to keep the plants warm even when the temperatures are cold outside of the greenhouse. So we know that the solar radiation is transmitted through the glass, absorbed by the plants. It can be re-radiated out, bouncing around within the greenhouse and reflected within there, but ultimately the radiation is trapped within there, so then increases the temperature inside the enclosed space. So now some examples that we have in the plant world for this microgreenhouse is hollow stems, the loosewort flower, which is the yellow flower on the top, and the campion flower, which is the pink flower at the bottom. Ground cherries or Physalis fruits, that's the calyx that forms around the fruit and keeps it safe and warm as it's developing. And as well, at the top of the image, we see the plant called the Himalayan glasshouse plant. So this plant is actually named after this phenomenon that we're discussing, which is really interesting. It's very intuitive, but this hasn't actually been shown in the literature before. One example from Peter's work up in the Arctic is the calyx of the Arctic campions. And these are really beautiful flowers. They almost look like a little lantern. They're called Melangium epithelium. And the calyx forms a translucent enclosure. And the reproductive organs, the pistils within, absorb solar heat. And the enclosure warms to about 4 to 5 degrees warmer than the ambient air in the sun. But if the sun is not shining, the flowers are the same temperature as the air. So we can see that it's probably the solar radiation that is causing this temperature increase. Okay, sorry. So the next example we have are the Arctic pedicularis species. Now you can actually see in the photo the sun shining through the flower. You can see that it's a translucent material, and almost get an idea for the texture and the feel of the flower looking at that image. And in the center, this graph shows how the temperature changes when the flower is facing the sun, or if the flower is lateral to the sun, or the back to the sun. So you can see that the highest temperature differences are almost 7 degrees again, and the highest differences occur when they're lateral to the sun. When the sun is vertical above, yeah, this is what that occurs. Another example of a microgreenhouse could be snapdragon flowers that they open by a snap mechanism. They sort of open and close like this, but they are fully closed most of the time unless a pollinator is trying to get in there, or a researcher. So the temperature excesses within these flowers that we recorded was up to 7 degrees Celsius. Again, a significant difference, especially when you're looking at the colder temperatures. So question for you. Should the pale colored flowers be warmer from a greater microgreenhouse effect? More radiation is able to get into the center of the flower. It's not reflected by the color. Or should darker flowers be warmer due to more solar absorption of the petals than radiating that heat within? I'll give you a moment to think about that. Well, our results were inconclusive. We observed higher temperatures in different floral colors grown right next to each other in the same greenhouse, but there were no clear patterns that were identifiable with the results we collected so far. So there's a lot to still be left conceptualized about this question and about how microgreenhouses work and under the different conditions that they could be working. We do have some results from microgreenhouse heating within hollow stems, and these studies are fairly new to science. Peter was making some groundbreaking observations during his PhD, and then he's went back to work on this, and so far we're having really interesting results. So we see in balsam weed stems, they can be up to 3 to 4 degrees warmer than the ambient air. And you can see that they're a fairly thin-walled stem. You could definitely tell that the light is going to pass through there. It's not a woody structure, it's herbaceous, it's nice and soft plant tissue. Arctic mastodon flowers, another Arctic species. So you can see when you're thinking about in an ecological sense where the flowers might benefit more from having hollow structures. We see a lot of them in the Arctic, so that is very interesting. And Peter observed that these have growth of greater than 2 centimeters per day. And again, in the Arctic region, so these are low temperatures, but they're able to achieve very high growth rates. So could that be due to the fact that inside the stems, it's 3 to 5 degrees warmer than the outside air? Well, in our surveys that we've done so far in our lab from 2017, actually to 2020 now, in Ontario, in Quebec, and Manitoba, so different provinces throughout Canada, and also in the United Kingdom, we found that about 50% of plants have hollow stems. So this is a very widespread phenomenon that has not been studied in the literature, which is very interesting, as a very diverse amount of plants have hollow stems. So but the only theories that are out there about why certain plants have hollow stems is more to do with the structural integrity, that they don't need to be as strong like a tree, so they can afford to have a little bit less structure, but we think that there might be a little bit more to it than that. So next let's talk about some more examples of our hollow stem work. So Gerbera daisies, or Gerbera jamesoni flowers, they're one of the top 5 cut flowers in the world. So there's a very large commercial industry for these flowers, and we have a greenhouse close by here where we've been able to do some research in their commercial greenhouses. And you can see that Gerberas are also disc flowers. So in the images I have shown, you can see that the flowers, again, are warmer than the leaves. And you can also pick out that the stems are also warmer than the leaves. So we observed the higher than air floral temperatures in Gerberas. We found that the average surface temperature of flowers from this snapshot was 22.5 degrees. The average surface temperature of the stems was 20.7. And the average surface temperature of the leaves was the lowest at 20.1. Now, Gerberas are very interesting because they have diverse stem types. So some varieties are solid, and some are hollow, and some are somewhere in between. Now, Gerberas have been bred to have a variety of different flower appearances, flower colors, and even different layers of petals, but they're all about the same. So it's very interesting that the stems have evolved such different phenotypes. Now, the Gerbera growers, they don't like the hollow stem varieties because they're more prone to bending and snapping. So the cut flower producers, they don't like the hollow stems. So our research is in understanding what role temperature might play and how the temperature within the stems could then influence the floral maturation rates, or the floral, like how long the lifespan is, the shelf life of the flower. The hollowness of the stem has a lot to do with these different factors. So the more we can understand about why certain varieties have different hollow stems and how these hollow stems perform in comparison to the solid stems, that could really help out the growers. So here's some pictures from the Gerbera greenhouse. On the left, you can see a fine wire. It's actually slightly out of focus, but it's a pink wire going into the stem of an immature flower. So we're trying to track the temperature regimes from the development of the flower until when it blooms. Then we have a perfectly blooming flower that we were looking at, the toast variety. And this was a variety that was particularly interesting because the stem type was not consistent. Some of the stems within this exact variety were more hollow or more solid than others. And we've also read in the literature that this can actually be dependent on the time of year, is that a variety can have solid stems at one time of the year and hollow at another time. So I'll be continuing this field work into this fall and winter, and comparing it to last year and seeing some of the differences that might exist. So the questions that we're asking for the Gerbera study are, how does the stem type affect the internal temperature? And then also, how does the stem type affect the growth rate, floral maturation, and shelf life of the cut flowers? And on the right, you can see our methods. We have these plastic boxes where we have data loggers with thermocouples in them that are connected to the plants that are measuring the temperatures inside. Further back, you can see it looks like a little bottle on a stick. That's actually measuring how much light is coming in through the greenhouse, so we can see if it's daytime or nighttime, and then see at which light conditions the temperatures have the highest difference between the flower and the air. So some of our results so far, we can see these were two of the varieties that we were looking at for most of our research. The top variety is pink, called Rendezvous, and the bottom yellow and orange is called Toast. Now we found that the hollow stems reached both hotter and colder temperatures than the solid stems that were grown side by side. We also found that hollow Gerbera stems reached their maximum temperature differences during daylight hours under sunny conditions. So this is evidence for the microgreenhouse effect happening within Gerbera stems. Another example that we have studied at the University of Guelph is Paperwhite Narcissus stems. We found that these hollow stems can be about 5 degrees warmer than the surrounding air. Now these stems are hollow and translucent, and you can also see on the top left corner that the inside surface is very shiny, but it's not wet. So it looks like it could be a microgreenhouse effect with this light coming in and then becoming trapped, coupled with a thermos bottle effect, because the shiny outer appearance acts like tinfoil or like a thermos to keep all the heat reflected inside, like a mirror. So this is something that we've observed within certain hollow stems that have this very shiny texture to them that could be increasing the reflection rates on the inside of the stem. The next study plant that we've looked at is pumpkin. So pumpkin is another very popular species to grow here in Ontario. So we've had access to a Strom squash farm where Patricia and I first started the research a couple years ago, and I was able to continue with some of that this summer. So we can see on the left top of the screen, again, the plastic box has the data logger in it, and these plants are still in flowering. So we had thermocouples measuring the temperatures on the inside of the stem, and the little thing on the stick is a radiation shield to have one that's have one air temperature that's also shielded from the sun to see how that compares to the air temperatures that are just left out. You can see the developing pumpkins, and we found that inside the stems, they can reach up to 10 degrees warmer than the air temperature. So this is very significant. And then on the right, you can see the texture of the stem. It has somewhat little spikes on it, so it could be increasing the boundary layer, somewhat like how the fuzzy or the hairy flowers are, as well as the texture of the tissue is very thin to allow a lot of light to pass through. We found that the highest temperature differences occur at the highest amount of sunlight in the day. So another very interesting thing that we were able to see this summer was using the spectrometer to see which wavelengths of light are present inside the stem. So this is the image at the bottom of the screen, the graph. You can see there's a dip in the red light, and so this is makes sense because we know that red light is mostly used for photosynthesis, and there's a peak in the green spectrum because we know that a lot of the green light is reflected, and that's why plants appear green. So in doing this method, we can compare and see if certain varieties have more or less light that's let into them, and how that spectra compares to the sunlight, and see if there's any patterns that we can see there. So a short summary of some of the other stems that we have data on. The temperature excesses in the sun in Paperwhites were up to 5.8, or sorry, the mean was 5.8 with a standard deviation of 0.7 degrees. And in cloudy weather, it was only 1.15 degrees. In daffodils, the temperature excess in the sun of the flower was up to 6.6 degrees. But if there was cloudy weather or at nighttime, it went only up to 2.7 degrees warmer than the air, or actually down colder than the air temperature, below 0.2 degrees Celsius below the air temperature. Japanese knotweed is another example. This is a very invasive species we have here in Ontario. So the more we can know about it and how it grows and how its relationship to the environment, the more we can know about how to control this plant. So we found that Japanese knotweed, the temperature excesses are up to 4.6 degrees, and they actually get significantly cooler at night or in cloudy weather. And now another interesting aspect to this project is studying how the microgreenhouse effect could also affect hollow fruits and help heat them up in order to reach their maturation or their seed dispersal strategy. So these again are some new studies to science, and I'll talk a bit about each of these species of plants shown above. They all have very interesting body plans, and there's a lot of diversity in this group. So the first one is wild cucumber, or Echinocystis lobata. So we can see on the right hand of the screen the spiky ball. It has a few wires that are, one is measuring the air temperature and one is inside inserted. So we can see that in direct sunlight, delta T, the temperature of the fruit minus the air temperature, was above 8 degrees, and that was the mean for this group. So that's a very significant result. And in cloudy weather, the fruits were actually lower than the ambient air temperature. And we can see here on the left picture the seeds inside of the fruit, and the fruit has not been cut open. This is actually how the seeds are dispersed. It explodes, and then the seeds fall out of it. So we think that because of the spongy outside casing around the seeds, that a lot of heat is being trapped within this insulated zone, and it's also translucent. So a lot of heat is being trapped in there, thus maturing the seeds on the inside and allowing for this bursting to occur. Now here we can see some of the surface temperatures of the cucumbers. We see on the right image, there's a few little balls of light, and on the left of the image, you can see a few of those cucumbers, and you can see that those are that they line up. So we can see that the fruits are quite a lot warmer than the air and warmer than the leaves as well. The next plant is Physalis. So this is very tasty fruit, ground cherries or like a tomatillo. And we found that within the calyx, you can see some of the wires being inserted in the top left. They're up to 6.6 degrees warmer than the air in direct sunlight. And on the right, the graph is showing, with the blue line, is showing the temperature of the fruit and the ambient temperature. And now this is 3 days of recording that we are showing here, from May 17th to May 19th. So each peak represents one night and one day, and then night and then day, and then night and then day, and then night again. So this daily temperature log of the developing fruits, we can see that the fruit temperature reaches much higher temperatures than the air, from about 8 a.m. to about 6 p.m. So during the daylight. And on the middle day, the temperatures were more steady, and that day was actually more cloudy than the day previous and the next day. So you can see when it's sunny out, the effect is greatest. Okay. Next, we have milkweed, which is similar to the or somewhat of a similar structure to the wild cucumber, although this is a follicle and not a fruit. The milkweed, as it develops, the left-hand side is a very young pod where the seeds are not formed fully on the inside. It's just kind of this network of pre-seed material, and then you can see on the middle image when the pods have burst, and then the seeds are able to disperse to the wind. Now, we found that these are up to 13 degrees warmer in direct sunlight, and I think that's the highest temperature difference that I've shown in this entire presentation. So this is quite significant. Double-digit difference between the air and the fruit temperature. So this phenomenon could be due to the microgreenhouse effect, and this effect is necessary for this plant to develop, as before we're talking about the seed dispersal mechanism, in order for the plant tissue to dry out and burst as the way that it does. The heat would have a component in allowing that to happen and allowing the material to dry out more, and then it can have this bursting mechanism. So we were able to study the milkweed just at the parking lot at the University of Guelph. And milkweed is a very ecologically important plant, as it is the primary food source for monarch butterflies. So this plant is, from a conservation perspective, is a very important plant. So in order to understand how this plant reproduces, I think it's important to consider the microgreenhouse effect. Another very beautiful plant that we have studied is the nigella, or black cumin, and within the seed heads of the flower, we found mean temperature excesses of 5.5 degrees, plus or minus 0.4 degrees Celsius in sunlight. Again, the seed bursting mechanism exists in this plant. So high temperatures, could contribute to the drying of the cell wall and the development of the seeds. As you can see, the top, where like the fingers are, they burst and fold out in this beautiful formation, and the seeds are able to fall away and spread. Okay, so now that we've shown some of the examples of the microgreenhouse effect, we can discuss a little bit in more complex detail all of the different heat pathways that could occur. So, firstly, solar radiation. So the solar radiation comes in from the sun. The letter A is showing the solar radiation in this diagram. So when the sun hits the wall, it could be, number one, reflected off. Number two, absorbed within the plant wall. Or some could be transmitted through the plant wall. And then after some of that, then we'll go on to absorbed radiation. So absorbed radiation within the plant cell wall can then go through, can be lost into the stem or out of the stem. It can be transmitted / conducted into the stem or across all the way to the other cell wall, or sorry, plant wall. This isn't a cell, this is a larger structure. We're looking at the entire stem. And when it's transmitted and conducted, it then can add energy into the lumen, which is the space within the cell. Within some that has been conducted, the radiated energy can also then be reflected and absorbed again. So this is number 5 and number 6 shown in the diagram. If the radiation is then absorbed by atmosphere in the lumen, so the air within the stem, number 7, then another process of heat exchange could occur by conduction, by like direct contact, convection, which is the airflow that we were describing earlier, or radiation. So we know that conduction, convection, and radiation are the 3 ways that heat can travel. So all of these play a role within the microgreenhouse effect. And now this is a very complex situation, and it would be very hard to study each heat pathway. So we're not going to be able to do that with our results that I've shown today, but hopefully by the end of my master's, we'll be able to have a more mathematical version of this model where we can make predictions about, you know, if there's this much radiation and the plant wall has the capability to transmit so much radiation, then we'll be able to know a lot more about this mechanism and be able to fill in some of these numbers with actual equations. So all of this together creates the greenhouse effect. The heat in the stems can also then be liberated into the environment by radiation emissivity. So that's like number 3 on this diagram. And it could also be conducted out into the environment if something is touching it or the air touching it next to the stem. There's some conduction that can occur, and it can also be conducted up and down the stem. So this is another large factor to consider, is how the heat will flow through the stem and can flow up into the flower organs, down into the roots. And this is conducted through, of course, the phloem and the xylem, shown in number 2LO along the side of the plant wall. And then some of this heat is also used in photosynthesis and metabolism, which we're not showing here. But this image does show most of the mechanisms that we know must be occurring. But nonetheless, even though there's all these pathways for heat to escape the stem, ultimately, we are seeing that the heat in stems is higher than the air, so we know that a lot of these mechanisms are occurring, that the heat is being reflected inside, and the radiation is being trapped inside the stem, as we have shown that this effect only occurs when it's sunny. If there's no sun, then these mechanisms do not come into play as much. So now I'm going to go a bit more into detail about some of our methodology and the mechanisms of heating in enclosed flowers, fruits, and stems. So I have talked about a lot of this throughout the presentation already, but just to put it all together. So we use a lot of temperature measurements. We're using thermocouples. The wires shown here, they're blue, and thermoresistors is another option for this. And this data logger, this is one that Peter is using. It's just a handheld one. You can take it out in the field. It's very handy. We also have ones that will record, they'll record for long periods of time, weeks at a time, that we can leave out in the field with us. Now we also take a lot of measurements of the stems, of the shape and the form. We observe the physical dimensions of the plant. And as I showed before, we are using some spectrophotometry, looking at the reflectance and the absorbance and transmittance of wavelengths of light, including the infrared light. And we would like to get more into histology, morphology of the cellular level and the subcellular level, and how the cell structure actually could be contributing to these processes. And unfortunately, because of COVID, this past summer, we weren't able to do much with the microscopes because we weren't really allowed on campus. But because I was able to do some field work, I'm hoping that this part of the project can still go forward in the future. Yes, again, we wanted to do light and electron microscopy to get really high quality cellular morphology results, but as this equipment is very expensive, we haven't been able to do this yet. One thing that we have been able to do is the IR thermography, which is the infrared images that I've been showing, and this has been a really good technique for us to to use, and when we're seeing something that we think is occurring, then we're able to both record it with the thermocouple and also with the infrared camera, and then we're able to see, yes, this is a phenomenon going on here. So this slide basically shows which characteristics are present in different aspects of the plants. So this is a synthesis of the state of the science and how interdisciplinary opportunities in biophysics exist. So if you're interested in looking at pigments and cellular pigments, this characteristic is present in flowers, stems, and fruits. Where the pigment is placed within the cell, again, within flower stems and fruits, but this is less studied. Pigment absorption and reflection, this is highly studied within flowers and more so in stems, not as much in fruits. In the morphology of the flowers, stems, and fruits is highly studied in flowers, but as I mentioned before, the stem morphology is not really well understood. Even in the example that I was using of Gerber growers, they know that these different stem types exist, but they don't know why. And in fruits, this morphology in terms of biophysics has not really been studied. On the microscopic level of the morphology, we have more studies again happening in flowers and stems than in fruits. And the study of color or colorimetry in floral and stem and fruit science, of course, again, mostly in flowers, as we know that flowers can attract pollinators, so this is more highly studied in flowers. Now, the floral behavior, the following of the sun, the diheliotropism and epinastism, this has been studied in flowers. In stems and fruits, not as much. Even though the stem is a part of that, following the sun, it hasn't really been looked at. But it would be interesting to see if, like a dandelion, for instance, has a hollow stem, so maybe the stem is also interacting differently with the environment due to the following of the sun behavior. Now, internal temperatures of plants has somewhat been studied in flowers, as we were showing earlier today, but the internal temperatures of stems and fruits has not been studied very much in the literature. So this is a large gap in the research. And the mechanisms for the internal temperature regulations has only been studied in flowers again. So the mechanisms that we were talking earlier about the different shapes of flowers, this has been well studied since the 1700s, but the mechanisms for knowing, for example, that cucumbers are warmer, this has only been able to be known recently because of the technology advancements and because of the research interest that is there. So hopefully going forward, we can start to fill in some of these areas on this chart to try to understand better plants and how they interact with their environment. We know, given climate change, that knowing as much as possible about how plants interact with their environment, given the change in climate, that's going to really help us to develop conservation strategies, as well as focus on sustainable production of the plants that we know to be microgreenhouses. This is, yes, okay. So some of the relation to pollination and plant reproduction that we see is either in general botany or botanical specifics. So in general botany, temperatures affect how the flower actually matures. So how the anther matures, how the pistil matures, how both the male and the female mature, and how the pollen tube growth actually occurs when the pollen lands, and how the tube growth occurs. The temperature can have an influence on all these things. The temperature can also influence the floral display of how visible or how conspicuous, if it's hiding, if it's closed. So this temperature can trigger the opening and closing of flowers. Temperature also has a large relationship to how pollinators interact with the flowers. So what we saw in the Arctic flowers is that a lot of these flowers, although they're protecting their own reproductive organs, they're also enticing the pollinators to come to them as a warm spot to get warm, and so then they can then fly to the next flower in the cold air. But when they're in the flower, they're nice and warm and protected. So this is an enticing mechanism to get pollinators to visit the flower. And now the temperature will also influence the nectar and how viscous, how thick it is, and also how warm it could be. So how delicious it is to the bees. It could depend on the temperature. And also the pollen display. So the pollen granules, they may be different under different temperatures. The seed and fruit display of a plant is also affected by temperature. So how is it affected by temperature? Well, the stem elongation is definitely a factor. The dispersal by wind and animal dispersal, these are all influenced by temperature to some degree. Now for the botanical specifics, we have seed production, propagation of plants, so how they're able to actually reproduce and how they're able to grow and form new plants. The floral display is also influenced by the temperature. So stem bending and floral shelf life, as I was discussing earlier with the Gerberas, the hollow stems present a problem with the stem bending. How the temperature comes into play, well, we know that hollow stems have higher extreme temperatures, and therefore they could have an influence on the shelf life, on the floral life of the flower, as it has reached more significantly extreme temperatures during its lifespan than a solid flower, so it might have different characteristics at the end of the day. Stem elongation can also be related to temperature fluctuations. So there's a lot of evidence that shows that if plants are exposed to temperature fluctuations or light fluctuations, then they will actually increase their stem lengths because they're growing towards the sun or going towards the light. If they're shaded for some part of the day, then they could increase their stem length because they're looking for that light more. So stem elongation can be paired with temperature fluctuations. Temperature is also, of course, related to water relations and the transpiration of the plant. So as we were talking about before, is that when the heat is trapped within the stem, and then it could be conducted up and down the stem, and there's also transpiration that's occurring from the leaves and from the flowers. So some of that heat is going to be lost through transpiration, and maybe if the plant is heating up more, there's more transpiration happening, so there could be more irrigation, more need to have water put back into the plant, put back into the soil. So to understand water relations and water conservation, to understand the different needs a plant might have, if it's hollow or solid stemmed, that's very important. Now, diseases, of course, can also be affected by temperature. Fungal and bacterial growth on plants, such as mold, could increase under hotter temperatures, under higher humidity. Now, this is something that growers always are trying to prevent disease on their plants. So if the connection between hollowness and the microgreenhouse effect in certain diseases was expanded on, this could help in coming up with disease prevention or disease control ideas. Sorry, I reformatted this slide. This is the same as the other one. I forgot to take out the old one. So this is the same as the last slide. We're looking at the general and botanical specifics, but this one I just included some pretty pictures in. So there's nothing really else for me to add at this point. So thank you very much. And I will take it to the chat for any questions.
Thank you, Charlotte. It was very nice presentation. Thank you very much. I'm very glad to hear all the progress that you have done since I left Canada. It's amazing. And Larissa is a professor from our department, and she's the coordinator of the graduate program. And she said that it was a fantastic talk, so many different ways to regulate temperature in plants. My favorite was in bellflowers, and she has a question that you and Peter can answer. So if you know if this higher temperature also occurs in hollow stems in tropical regions.
Peter, do you have a response for that question?
Well, we know one of the plants with hollow stems that grows the fastest in tropical regions is bamboo. But bamboo stems, generally speaking, are not translucent, and so we have no idea what goes on in terms of the temperature inside bamboo stems. We do know that bamboo stems have a different gas composition to the outside atmosphere. But as for tropical plants in general, we don't know very much about what the proportion of the flora is of herbaceous plants that have hollow stems. This is unknown, and certainly we're asking anybody in the world to send us information on any particular plant that they find, give us a little information on its habitat and what it looks like, and maybe a photograph, and then whether the stem is hollow or not. And maybe over the years we'll be able to generate a world atlas of hollowness of stems. But it is surprising that nobody has reviewed this in a general sense, a taxonomic sense, an ecological sense. It's just at the moment a fairly open question. And as Patricia mentioned in the talk, we have found in the temperate regions of North America and of the United Kingdom, where 60–70% of the plants, herbaceous plants, have hollow stems. So this is quite a lot of a higher proportion than really I expected, but I had no reason for expectation apart from just, sort of being a keen naturalist, going out and looking at plants in the general environment. So yes, information from the tropics would be very useful. Would be interesting to put into the sort of general equation. I can see there should be no reason why there shouldn't be a lot of hollow-stemmed plants in the tropics.
Yes, Peter, Larissa sent me a message and said that you two should come to visit us and work on this here in Brazil, and I agree. Yes, actually this year, in the beginning of 2019, we had a field course, and I took the thermometer that I brought from Canada, but I didn't find any herbaceous species with hollow stems. So I was looking at this. Peter knows this place, Promata. He has been there for the pollination course. So I went walking on Promata and cutting stems, but I couldn't find any. But one of them we could, I won't remember the name of the plant. Some students did some measurements, but the stem wasn't, it wasn't live tissue. I guess it was more dead tissue. So they couldn't find any difference between inside and outside temperature. And I have a question. So do you expect to find this difference in temperature only for plants that the stem is alive, like for live tissue, or dead would have this this effect also?
Well, I could try to answer this question. I think that the effect would be different with dead tissue because the tissue would reflect and absorb differently based on what we know about the pigments and like what I showed with the green and the reds being absorbed because of photosynthesis. So I think that the lights being, the wavelengths being transmitted would be different, but I think the effect could still take place because if it was just some radiation still able to transmit, then if the material was still enclosed, I think it would heat up, but we haven't studied this very much.
Yeah, I would think that way too.
Because I think if we tried an experiment just putting like a plastic tube or any different type of material, you would see some sort of difference, but it would be dependent on the material. What do you think, Peter?
Yes, I think that would be correct. It would certainly. I would think that dead canes of plants, basically that are dead and just have gone brown or yellow, would pass light through them, and there would be a greenhouse effect there. It would be interesting from a point of view of habitat for insects that live in such places to know how important it is. But we don't know of any work that's gone along that way. We have our minds filled with the complexities of thermodynamics in hollow structures that are alive without worrying about the other sorts of things. So there's a lot to be done. I think a lot of new things, new ideas to be explored, and I think that's the exciting thing. And as I say, it's very exciting that nobody really seems to have paid much attention to these phenomena, and we seem to be amongst the first to do this. In the world. So we would encourage students who are looking for small projects to do for a thesis or a term paper or a small project to do for handing in, to look into this and see what they can find out on their own and discover the extent of this seemingly novel to human beings phenomenon. Obviously, it's not novel to nature. It's been even the horsetails, the Equisetum species, have are hollow, and presumably exhibit these microgreenhouse effects, even though they have been around on the surface of the planet for millions and millions of years. So evolutionarily, it's not new, but intellectually, it seems to be unknown.
Yeah, that's nice. We don't have more questions. I guess people are shy because of the English, and but I think it was a great talk and a lot of information on it. And we have to talk about our paper now. And I'm very glad to see you both. Like it's been some time that I don't see you. guys. And thank you very much for being here with us today.
Yeah, you're welcome. Thank you so much. I would add just to your question about, I was thinking a little bit more about your question of the dead plant material. Now, in our survey work, we have found a lot of plants that are solid at the start of the year, and by the end of the year, they become hollow. So it would be very interesting to look at that process more in depth of like this, you know, transferring from the live tissue to the dead tissue. What are the processes that allow that to happen, to the hollowing out of the structure in the middle, and maybe there's some interesting phenomenons there. But yeah, that is one thing we've observed, that some flowers will be solid at the start of the year, and then we go to them at the end, they're hollow. So it is quite interesting. Yeah.
Well, someone just sent a question.
Okay.
So, thinking about a future higher global temperature scenario, do these plants have a mechanism to avoid overheating?
Well, we know that transpiration is a mechanism to avoid overheating, where the stomata open and close to allow the excess moisture and excess heat that's produced because of photosynthesis. So we know that that exists. I would think that under higher temperatures, we could see that hollow stem plants perform better, although this is a speculation, just because we know that they already reach more extreme temperatures than the solid counterparts, so they could be able to handle the higher air temperatures as well, and then have the other extremes. But this is unknown. We haven't tested any of our plants under different conditions of like increasing the temperature artificially and then seeing how they react. So I'm not able to fully answer that right now, but what do you think, Peter?
It's a very, very interesting question, and one that if one thinks about it, it sort of goes both ways. There are a number of issues associated, particularly that have been noted in temperate and Arctic areas, where increasing global temperatures or increasing temperatures are resulting in mismatches in the phenology of the production of the flowers with respect to the production and availability of pollinators. And presumably that carries right the way through to the production of seeds. But only recently have people started to look at those phenomena, and they have not been looking at the additional heat gain inside plants as a result of microgreenhouse effects, for example. It's pretty much an open area for anybody to really sort of think about and come up with some hypotheses which would be worth testing. I think that one place in Brazil that would be very interesting to look for these hollow structures would be in the Chapada da Diamantina. And there are a lot of herbaceous plants there. They're fairly low growing. And it may be that in those sorts of locations, there could be some very interesting phenomena. Again, because at certain times of the year and certainly at night, it can get quite cold. And then during the day, there's a lot of intense sunshine, and that's, we presume, would contribute to the growing of all the very interesting plants of the Chapada. So that would be an interesting area to look at and to survey.
Yeah, that's true.
Tomato too, of course, because that's can get quite chilly up in tomato.
Yes, it can, and suddenly. Yes, last January we were there, and it was cold at some points at night, at least. So I would expect to find some plants with hollow stems in Rio Grande do Sul, in southern Brazil as well. But I think we have to look for more for them. Yes.
I think another aspect to this question is also, it depends on the plants that you're talking about, if it's going to do better under hotter or colder conditions. A lot of the research that I've been looking at in the literature that talks about surface temperature of plants actually looks at the other end of the spectrum, or that frost damage. So they show that the plants will reach colder temperatures. A variety of different crop plants reach colder temperatures, and that the temperature of the leaf is actually what is determined if damage occurs rather than the temperature of the air. So I think it's just the knowledge of us knowing that plants don't always take on the air temperature, and that we're able to see what the surface temperature of the plants are using our thermometers or using our images. Then we can then see, okay, well, if disease sets in at this level of a certain mold, or frost damage occurs at this level of surface temperature of the plant, then we're that much closer to knowing, how it's going to react under different conditions. If we can know what the true temperature of the plant is, that's the first step to knowing how it will react under more extreme conditions.
Wow.
Does that make sense?
Yes, it does. Well, I guess we are now at 7 p.m. here, and it's when we usually end the session.
Perfect.
So I will thank you again, both of you, for joining us today. And I hope I see you guys soon.
Yes, I hope so too.
If I were there, I would take everybody for some cervezas.
Yes. Yes, we have to wait for the COVID to go away, coronavirus to go away, because here is not so simple, but everything is open, so we could go to drink beer, to drink cervejas. Okay, and they will send me the recording of this presentation.
Okay.
And usually I upload it on our YouTube channel, but I can send you also it.
Excellent. I appreciate that. Thank you.
Okay. Have a good night,
Yes, have a good
everyone.
night. Bye.
Bye.
Tchau,
Muito
gente.
obrigado a todos.
Muito obrigada, Peter.
Bye.
Bye.